Okay, everybody, I am sharing screen. What we are studying, ultrastructure of prokaryotic cell. What do you mean by ultrastructure? Ultrastructure means internal structure. Okay. Now, in this picture, you see a bacteria. Okay. And this bacteria's internal structure we are going to study. Different parts of bacteria we are going to study. Okay. Now see, when we study any internal structure, okay, particularly prokaryotic cell internal structure, prokaryotic cell internal structure is divided into two parts basically. Okay, for studying purpose, structure, two parts we can study. Okay, first part is something called as cell envelope. First part is called as cell envelope. Cell envelope, and second part is second part is what to say, cytoplasm. Cytoplasm. Now when I first we will study cell envelope. Okay, now when I say cell envelope, means outer covering of the cell. Outer covering of cell. So, cell envelope, that is outer covering in bacteria, made up of three different structures. Okay, made up of three different structures. Cell envelope made up of three different structures. Outermost, this red colored, which you see in this particular diagram, red colored. Okay, it is called as the capsule. It's called as what? Capsule. The second, that is here you see yellow color, it is cell wall, right? Cell wall. And third structure is plasma membrane. Plasma membrane. All these three structures together is termed as cell envelope, right? Okay. All these three structures together called as cell envelope. Right? Complete this writing, then we'll come out of screen and then I'll teach you about capsule first, then cell wall, and then plasma. Completed?
Done, ma'am.
Done. I'm now coming out of screen. See, now consider bacteria, very small, microscopic structure. Okay. And it is having the outermost covering, it is called as the capsule. Now remember whether bacteria can live in two environments. Okay. Some bacteria are living in water, liquid environment. Okay. So liquid environment, you can imagine different liquid environments. See, bacteria may live in, first of all, I try to understand this. Bacteria can live anywhere in the world. Okay. They are, if you studied bacteria like in your 8th, 9th standard, I don't know, have you studied or not, but they are called as omnipresent. Omnipresent का मतलब है we find them everywhere. We find them everywhere. Okay, so we find them in ocean. Okay, they can live in air. They can live in fresh water. They can live in sea water. They can live in your food. They can live in snow. They can live in they can live in boiling water. They can live anywhere. Understanding? So any environment you imagine, bacteria will be living over there. Okay, they even live inside our body. Okay, on my skin right now, suppose we did like little testing and observed it under microscope, we will find thousands of bacteria on our skin. Okay, we may find thousands or millions of bacteria inside our intestine. Okay. So there are so many bacteria living everywhere. Okay. So the capsule, which is the outermost covering of their body, provide them protection. Okay. Provide them protection because they can live anywhere. Okay, they can live anywhere. That's why that Lysol and all advertise. If you see, it kills 99% of bacteria. Correct? Why it killing 99% of bacteria? Because they are there. Okay, they are there to kill. Okay, let it be. So if on this table, on your computer, on my computer, there are so many of them. Okay, but remember, these people know in advertise, they advertise bad about bacteria. Okay, they say bacteria are bad, but not all bacteria are bad. Okay, very few bacteria are disease causing. बाकी के कुछ नहीं करते. They don't do anything to us. They don't bother human exist. Okay, they had nothing to do with humans. Okay, they are not there to kill us. They are just there, living there. We need not to kill them. Okay. Only very few bacteria are basically disease causing. Very few of them. Some causes typhoid, some causes cholera, some causes tuberculosis. Okay, but very few of them. Okay, so let's come back to our point. So ये जो outer covering होता है, the outer covering called as capsule provided protection. Now imagine right now the bacteria is living in water. Right now the bacteria is living in water. When the bacteria live in water, the capsule form a slimy covering. Okay, that capsule absorb water and form a slimy covering. One second. One second. Hello. Hello. Hello.
Yes.
Sorry. Actually, my brother went to Dubai and he don't get chance to call usually. And now he called, so I had to pick up. Okay. So, um, okay. So let's come back to the point. So what I was telling you, when the bacteria is living in water, this capsule take up water, absorb water, and become slimy. Okay. So suppose you pick up bacteria from the water, it will be slimy. And what thing is slimy? Its capsule part will be slimy. Understanding? And that slimy capsule will protect it from the watery environment. Right? The capsule, watery capsule, will protect bacteria into the, or slimy capsule, protect the bacteria in watery environment. But suppose that bacteria is in dry environment. It is in air right now, suppose. Okay. So the same capsule will lose its water. It will become like a hard covering. Right? It will become a hard covering, and that hard covering will protect it from the wind, from the air, dry environment from the air. Understand it? So capsule role is protection. When in water, it becomes slimy. When in dry environment, it becomes hard. Understanding? Okay. Capsule absorb water and become slimy. Right? Become slimy when in water, and when bacteria is in air, it is hard. Capsule become hard. Capsule is basically hard and protective. Right? Hard and protective. Then the second part is cell wall. Second part of the cell is cell envelope is cell wall. When I talk about cell wall, remember cell wall in bacteria is called is basically non-living part. Non-living part का मतलब होता है it do not non-living part means it do not repair itself. Understanding? It cannot repair itself. It cannot consume energy. It do not need food. It do not need energy. Understanding? Cell wall in bacteria is non-living part of the cell. Okay, non-living part of the bacteria. Plus, it is like what to say? It is also a protective covering. Its function is protective covering. Okay. When I talk about a cell wall as a protective covering, remember there are basically two different type of bacteria. Okay. Bacteria we can classify in two different types depending on cell wall. Okay. Bacteria maybe with cell wall. With cell wall is present. Cell wall present. Okay. And cell like bacteria may be without cell wall. Without cell wall. Here cell wall absent. Cell wall absent. Remember, almost all bacteria show presence of cell wall. Only mycoplasma without cell wall. Mycoplasma is the only group of bacteria which is without cell wall. Understanding? Remember this. Okay? Remember this very, very carefully. And when cell wall is present, there are again two groups of bacteria. One bacteria are called as archaebacteria. Archaebacteria. In another topic, we will learn about archaebacteria in detail, but afterward. Okay? Archaebacteria. Another bacteria called as eubacteria. Eubacteria. ये बैक्टीरिया के दो ग्रुप है. In one another topic, we will learn in detail about archaebacteria and we will learn detail about eubacteria. But right now, just try to understand. Archaebacteria are the one who live in harsh environmental conditions. Harsh live in harsh environmental conditions. Live in harsh environmental conditions. Mental conditions. Live in harsh environmental conditions. So what do you mean by harsh environmental conditions? Harsh environmental condition matlab they will live in boiling water. Okay, boiling hot water. Okay, boiling hot water is present in hot water spring. Do you understand what do you mean by hot water spring? Vaibhavi? Do you know?
Yes.
So hot water spring, okay, in hot water spring, they live in that boiling water, okay. They live in stomach of cattle, okay. गाय और बफेलो के स्टमक में रहते हैं, okay. Okay, harsh environmental condition. In a ruminant stomach, ruminant stomach is a special stomach cattle are having, okay, of cattle. That is, cattle means buffalo and cow, okay. So they live in such a harsh environmental condition. को archaebacteria कहते हैं, okay. In another topic we will study in detail about archaebacteria, but right now I just want you to understand, in archaebacteria, cell wall is made up of a chemical called as pseudo peptidoglycan. Okay, remember this name of chemical. Or eubacteria are the normal bacteria which live in normal environmental condition. And in this bacteria, cell wall is made up of peptidoglycan. Right? Remember, in archaebacteria, cell wall is made up of pseudopeptidoglycan, a chemical name as pseudopeptidoglycan. And in eubacteria, that is normal bacteria, cell wall is made up of peptidoglycan. Right? Clear?
Yes.
Then the next thing which you need to understand is that the third part of the cell envelope is plasma membrane. Is what?
Ma'am, can you tell up? I'm noting down.
Yeah. I thought you finished. Tell me when you complete it, okay? Three groups, make like first two groups, and then divide the first group into two more, okay? Shruti did not join only, no? Didn't reach home, I think.
Done, ma'am.
She did not check message even. Not reach home, I think. Okay. So, are you understanding? Vaibhavi?
Yes. Yes.
Now, next is plasma membrane. Remember, Vaibhavi, plasma membrane is even called as a cell membrane. Okay. And bacteria, eukaryotic cell, all plasma membrane is same. And in eukaryotic cell, I'll teach you everything about plasma membrane. Okay. There we have detailed study of plasma membrane. That time only we will study plasma membrane. Okay. I'm not teaching it right now. Okay. But this is the third covering. Remember, innermost is the plasma membrane. उसके ऊपर है cell wall, उसके ऊपर capsule. Right? Remember this. cell envelope. Okay. Now let's talk about the cytoplasm. Now about this cell envelope, I'm going to teach you one more technique called as gram staining. Gram staining. But this gram staining technique, no, you never studied before. So when Shrishti is there, no, that time only I'll teach you gram staining. Maybe tomorrow. Okay. Today I'll teach you simple stuff. Internal structure of bacteria we will study. Gram staining depend on the cell envelope, but that I will teach you tomorrow. It is a practical that you are going to do in exam. Okay. Not in exam actually, in practical, in your practical session. Okay. But question come on that part also. So we will study that. Understanding? Okay, so see, we studied about capsule, we studied about cell wall, we studied about plasma membrane. Now let's study what we see inside the bacterial cell. Okay, so when we see bacterial cell from the inside, okay, the most space is occupied by its genetic material. Most space is occupied. One second, we'll just copy this diagram down. So we will get enough space to write. So the most space inside the bacteria is occupied by this genetic material. That is its DNA. Genetic material. Okay. And remember, there is no nuclear membrane above the genetic material, correct? Genetic material without nuclear membrane. Right? Without nuclear membrane. So this genetic material without nuclear membrane is called as nucleoid. Remember this term. It's called as what? Nucleoid. Right? The nucleus without nuclear membrane is called as nucleoid. Okay? Remember this term, very, very important, nucleoid. Okay? Now, now suppose this genetic material is entangled. It is like a thread, correct? It is like, very much gathered together and compactly placed. Suppose I separated it, okay? What I will find is very large circular DNA. One very large circle of DNA, right? Large circular DNA, right? Circular DNA. So remember very, very carefully, the genetic material of bacteria is what? Large circular DNA. And this circular DNA only, they entangled, they just compactly packed and fit it. Understanding? Okay. Large sized circular DNA is what? Genetic material of bacteria. Is genetic material of bacteria. Remember one more thing. There is no histone protein. Right now you don't know what exactly is histone protein. That we will study in eukaryotic cell. But here right now just you remember no histone protein or histone protein absent. Just write what? Histone protein absent. Histone proteins absent. Okay. Fine. Then, so large space into the cytoplasm is occupied by this nucleoid, occupied by this small cell or very large size circular DNA. Understanding? Then there will be cytoplasm, liquid part. Then there will be liquid part. In that liquid part, we see a small bubble-like structures. Correct? Small these bubble-like structures. These are called as inclusion bodies. Inclusion bodies. Those are called as what? Inclusion bodies. Inclusion bodies store food material, store organic and inorganic food material. Mostly inorganic actually. Inorganic food material. Mostly inorganic, very few time organic. Okay. Food material. Clear? Clear about it? Then the next thing we and here are these ribosomes. दिख रहे? These are what? Ribosomes. Now ribosomes are small. बहुत सारे होते हैं बैक्टीरिया में. There are too many of them. Okay, small. They are small, non-membrane bound. Membrane bound cell organelles are absent. Correct? इनको membrane नहीं होती. Non-membrane bound cell organelles. Non-membrane bound, very important. Non-membrane bound structures I am writing. Cell organelle will make you confused. Non-membrane bound structure. Okay. Now every ribosome in bacteria, ribosomes are size of ribosome is measured in something called as Sedberg's unit. So is measured in Sedberg's unit. Sedberg's unit, and it is indicated as S. Right? It is indicated as capital S. So in bacteria, the size of ribosome is 70S. कितना होता है? 70S. 70 Sedberg's unit. Right? Clear? 70 Sedberg's unit, 70S. Okay. And what to say? This ribosome, every ribosome is made up of two sections. One is this and one is like this. It's shaped like this. The larger and whole size kitna hai? 70S. 70 Sedberg's unit. Right? This is called as a larger subunit. This one is smaller subunit. Right? Smaller subunit. Remember, the larger subunit is 50S और ये smaller subunit होता है 30S. Don't ऐसे नहीं होता कि 50 plus 30 is 80 ना, पर total size है 70. It's not the normal summation. Understanding? Okay. This is a size of bacterial ribosome. Clear?
Yes.
You wrote this? Do you want to note something down? Have you missed something? Have you wrote down about genetic material, nucleoid?
Yes.
Have you wrote histone protein absent?
Yes.
Inclusion bodies?
Yes.
Yes, no? Are we ribosome, small size, 70S, Sedberg's unit, everything?
Yes, ma'am.
So what we have studied till now? Cell envelope, first part kiya. Okay, correct? Cell envelope, this one, है ना? This one part we completed. Second part we completed about nucleoid. Third part we completed about inclusion bodies. Fourth part about ribosome, right? Fifth part little bit I did not write over here, but I told you about cytoplasm, correct? Cytoplasm is a liquid part present into the bacterial cell. Okay. Then the next thing present in the bacteria is called as something called as plasmid. The fourth structure is what? Plasmid. I will show you in the diagram plasmid, and then we will study about plasmid more. Okay. See, यहाँ है plasmid. Do you see that red colored small structure? Plasmid. You see that in diagram?
Yes.
So try to understand what exactly is the plasmid. Okay. So see. Plasmid. Now, Plasmid is very important from, like, it will come in like many topics. Okay, try to understand this very, very well. Okay, this is very basic knowledge you need to understand. By definition, what is plasmid? Try to understand. Plasmid is small circular, small circular, okay, self-replicating, extra chromosomal. Every term is important, okay? Extra chromosomal, DNA present in bacterial or prokaryotic cell. Present in bacterial cell, we also called it as a prokaryotic cell. See how I define plasmid? Plasmid is a small, circular, self-replicating, extra chromosomal DNA. Okay. So we need to know meaning of each and every word I wrote. Okay. First of all, you need to understand from this definition, plasmid is DNA. Plasmid is what? DNA. That is one important thing. Okay. Then it is a small DNA and circular DNA. Right? It is a small DNA and circular DNA. It is self-replicating. Self-replicating means one plasmid, suppose I have one plasmid, bacteria के अंदर एक ही plasmid है. After some time, it will create one more plasmid. Self-replicating means it can make copies of itself. Self-replicating is what? Can make copies of itself. Understanding? So after some time, and now I have two plasmids. After some time, this will create one more its copy. This will create one more its copy. So according to need, if right now bacteria need many plasmids, एक bacteria hundreds of plasmids prepare कर सकता है, right? Because plasmid create one more plasmid. Understanding? That property is called as a self-replicating. It can make its own copies. Understanding? So one bacteria, Vaibhavi, can maintain hundreds of plasmid, hundreds, literally. We will learn that in biotechnology more, but it can make hundred of copies of itself. Understanding? Okay. और एक्स्ट्रा क्रोमोसोमल का मतलब होता है, एक्स्ट्रा मीन्स outside the chromosome. Bacteria is having that large circular DNA as its chromosome, correct? Okay, outside the chromosome. So plasmid is a DNA which is located outside the chromosome, पर cytoplasm के inside, small circular self-replicating DNA. Understanding? Okay.
Okay.
And remember, Vaibhavi, only and only bacteria can maintain this. Okay, only and only bacteria can maintain this. In our body, we don't have any plasmid. Okay, we don't have any plasmid inside our cell. Okay, inside my body, I don't have plasmid. I don't have ability to maintain it. Okay, but bacteria have that. And bacteria को चाहिए क्यों plasmid? What it do with plasmid? Now remember one thing. Some bacteria have plasmid, some bacteria do not have plasmid. It depends on the type of bacteria. Okay, ऐसे नहीं है कि every bacteria have plasmid. No, some bacteria maintain plasmid, some does not maintain plasmid. Understand it? Okay. Now the bacteria which maintain plasmid, क्या करते हैं वो plasmid से? Okay. Now plasmid is not essential for the survival of bacteria. ऐसे नहीं है कि बैक्टीरिया मर जाएगा प्लाज्मिड नहीं है तो, नो, नथिंग. बट प्लाज्मिड help bacteria to do other work. Okay, means otherwise if plasmid is not there, those works will not done. Those work will remain like that. So for example, bacteria usually do asexual reproduction. One bacterial cell divide into two bacterial cells, that's it. But if bacteria decided to do sexual reproduction, it need plasmid. Understanding? Without help of plasmid, bacteria cannot do sexual reproduction. There is one other property called as antibiotic resistance. About antibiotic resistance, I'll teach you in another topic more. Okay. But there is one property called as antibiotic resistance. That antibiotic resistance also given by plasmid. Okay. If no plasmid, bacteria will not have antibiotic resistance. Understanding? That is also given by plasmid. So plasmids give very extraordinary properties to bacteria. Okay, bacteria can survive without plasmid, but if plasmid is there, it will have a superpower. Okay, it will have superpowers. Understanding? Easy?
Yes.
Okay, so we will stop over here today. Okay, I will upload this on YouTube for our Shruti, for our Shrishti. Okay, we'll see. If you also need, you can watch, but I will upload maybe tomorrow. Okay, I'll see when I get time to upload it. Okay, we'll stop recording.
Ma'am, you have YouTube channel.
I have.